This lecture will cover an overview of fungal identification, and it will include different lab methods and strategies for fungal identification. Mycology is the study of fungi, to include taxonomy, environmental impact, genetic and biochemical properties. Most fungi are septate, meaning that they live on dead and organic matter. Fungi can be yeast, or they can be molds. With yeasts, when they grow on a plate, they tend to be very moist looking. Molds will be filamentous, as in something like cotton, where there's filaments in it. And if you've ever had anything in your fridge growing with mold on it, then you kind of get an idea of what the filamentous look is. Organisms that display both yeast and a mold-like feature are considered to be dimorphic. They are thermally dimorphic, meaning that they are dependent on temperature as far as whether they're a yeast form or a mold form. Fungi that have more than one form or stage is considered to be polymorph. Let's first review what taxonomy is. So when we talk about taxonomy, we are actually referring to this chart over here to the side, which goes from life all the way down through species. So first we start with life, which is anything living, and then next we come to domain. And when we talk about domain, that is divided out with bacteria, archaea, and the eukaryota. So when we talk about the eukaryota, what we're focusing on for our next couple of weeks is the fungi. Now, within the kingdom of fungi, which is what we're talking about, the phylum is divided up into Zygomycota, Ascomycota, Basidiomycota, and Deuteromycota. Now, each of these are based on their reproduction, whether it's asexual or sexual. And the method of reproduction we will discuss a little bit more as we get further into this lesson. Now again, the reproductive forms can be telomorph, which is also known as a sexual form of reproduction, anamorph, or an asexual form, or cynomorph, which is different asexual forms in the same fungi. The overall working scheme of the fungi can be divided into yeast and mold. So if it is a yeast, it refers back to the yeast being moist versus mold being filamentous. The molds are further divided into aseptate hyphae and septate hyphae. So septate just means that it has a septum or some sort of wall separation within the cells. And the septate hyphae are further divided into the Demoticus and the hyaline. Hyaline is non-pigmented, whereas the Demoticus is a darkly pigmented type of mold. Here you can see that the fungi, the difference between the septate and the aseptate hyphae, and you can see the variations within each of these molds as far as this has divisions for the septate. It has a septum, so you can see these divisions throughout that wall, whereas the aseptate does not. And this is just another example where you can see the divisions. Now we talk about the different types of infections, then we're talking about the mycosis. So some of the various things that it can affect or invade the human host as a form of an infection would be a superficial cutaneous mycosis, which is an infection of the hair, the skin, the nails without any direct invasion of the deep tissue. So in other words, it's not going beyond the skin layer or the hair. It's not going deep into the tissue. And then you have the subcutaneous mycosa, which the infections are confined to the subcutaneous tissue without any deeper dissemination into your deep muscles and things like that. And then you have the systemic mycosis, in which the infection often involves the lungs, but it can disseminate into other organs throughout the body. And then you also have opportunistic mycosis, which is infections that occur in patients that have been immunocompromised, whether HIV or some sort of suppression to the immune system, like someone that may be being treated with high dose of steroids, or maybe a cancer patient that their immune system's been wiped out with chemotherapy. And these infections can become systemic. In other words, they can affect the entire body or any organ within the body. This chart here further divides out the different mycosis as far as the organisms that can cause that particular mycosis. For example, with cutaneous tinea, which is not to be confused with the worm that we just learned about, but this tinea is what is considered part of the infections that you may know as jock itch. Some people refer to as ringworm or athlete's foot. So we're going to learn about each of these and how they cause these types of infections. So when we talk about a fungi being able to infect a host, it's all based on some of the common virulence factors that those fungi have. Now this can be the size of the organism, its ability to grow at body temperature with a neutral pH, its ability to convert from a mycelial form. And when we talk about a mycelial form, we're talking about the classic form in which it takes on what we typically see growing in our refrigerators, or its ability to convert to a yeast form, which is the next column, or its ability to convert to a spirale form, which is right down here at the bottom, where it's kind of like a little cluster pod. And it also depends on its ability to produce toxins. Now, the type of pathogens that we refer to are primary pathogens and opportunistic pathogens. So if it's opportunistic, then that means that it's infecting someone who is immunocompromised. In other words, their immune system is not where it should be. Whereas a primary pathogen is going to infect someone that's immune system is working as it's expected. And it doesn't always mean that there's any sort of virulence factor coming into play, but it also may lead to some sort of subclinical disease. When you actually culture a specimen, meaning you put some of the specimen onto the agar plate, then the type of agar that you're going to use is going to be based a lot on what the specimen source is and what type of organisms you're looking for. So some labs may have a little bit of variation with what type of agar they prefer to use, but you will find a general consistency of the agar that is used for culturing fungal cultures. As far as incubation of those fungal cultures, they're typically kept at room temperature. Unless you're looking for a dimorphic type, then it could potentially be incubated at a different temperature. Most of the time, you will incubate these for 21 to 30 days with a humidity range of 40 to 50%. And so because you have that criteria, you do actually put these in some sort of incubator, something that looks similar to a refrigerator, except it holds room temperature and it regulates the humidity. And you have that humidity so that these plates do not dry out. Typically they will be examined 3 times a week, so a lot of labs will put it on a rotating schedule. For example, they may do it every Monday, Wednesday, Friday. It just depends on the lab and how many specimens they have coming through as far as to what their rotation is for how often they examine these plates for growth. So this is just some of the common medias that are used for fungal cultures. So you may see these with the pathogens when we start talking about the different pathogens of what they grow best on. For the Candida species, there are some plates that are differential. So they are made to distinguish one of the Candida yeast species from the other. And you will base how it grows on the plate by the morphology of the colonies and the color that it produces growing on those plates. You also have special media that will inhibit bacteria, so that if you have a specimen that could potentially be contaminated with bacteria, then you wouldn't want the bacteria interfering with the growth of the fungus. So you may have plates that would inhibit that bacterial growth. And you also have plates that are specific for particular types of fungus. So for example, the dermatophytes have a media that's specific for them to help with recovering the dermatophytes. Some of the direct detection methods within microscopy are potassium hydroxide, or what we refer to as KOH, the cauliflower white stain, and the Indy ink stain, and the lactophenol cotton blue. So depending on what you're looking for, and depending on what the lab's choice is, would depend on when you would use these particular reagents for microscopic viewing. The KOH is typically used with hair, skin, nails, fluids like vaginal fluids. This, when you mix the KOH with the specimen, then it will destroy anything that has the keratin in it, so any of your cellular components from the host. It will also destroy the hair, the skin, the nail. Depending on what you're testing, you may need to let it sit for a little while just to give it time to dissolve all of those. But once it's dissolved all of those, if there is the presence of any fungal elements, then you should be able to see those. And this is oftentimes used in doctors' offices when they are doing vaginal swabs. If they're looking for yeast growth, then they will use this to help visualize the yeast cells versus any kind of contamination through epithelial cells, normal skin cells, or white blood cells, red blood cells. So this is just an example of some of the different staining techniques used for microscopic viewing. The top one is the cauliflower stain, in which it fluoresces. The one in the middle is the Indy ink stain, which allows you to see the capsule around the yeast cell. And the one at the bottom is the lactophenol cotton blue, which helps you to see the different elements of the fungal species. When making stain slides of tissue, these are some of the common stains that are used, so you should be familiar with how the fungus would appear with the use of these particular stains. So these are some examples of how these stains would look. For example, here is the right stain with the organism inside of the white blood cell. This is an example of a PAS stain, And this bottom one is an example of a GMS stain. Now, laboratory diagnosis of various funguses can be done with serodiagnostics as well. This could be complement fixation, immunodiffusion, or enzyme immunoassay. There are also different molecular detection methods, and there's also the Maudie-Topf mass spectrophotometry. When identifying yeast, typically you will look at various things. First off, you like to see how it grows on the plate. Is it characteristic of a yeast? Then you would look at it to see what it looks like microscopically. Does it produce a germ tube? What types of sugars does it use? How does it react with those sugars? Does it produce urease? Does it have a capsule? So these are some of the things that we will learn when we are looking at the yeast and their characteristics. Mold as well goes through a series of things that can help you to identify the particular mold. You can look at it macroscopically, and when you look at it macroscopically, you are looking at it to see what it looks like growing on the plate. So not only are you looking at it, what it looks like at the surface of the plate, but you will also flip the plate over and see what it looks like on the underside of the plate. You'll note characteristics of color. Does it look fluffy? When you look at the microscopic characteristics, you will actually make a slide from the culture itself. So you would do this under a hood, and you would get some of the sample, and then you would put it onto a slide, and typically the lactophenylcotton blue is what you would use to look at its growth characteristics. So we talk about the colony morphology. Again, like I mentioned, you're looking at color. Does it appear green? Does it appear orange? And you will look at this on both sides of the plate. We also look at texture. Does it look like cotton? Is it real loose? Is it velvety? Does it have a granular look to it, or is it really woolly? Something like this green looking one here would be considered something that's kind of got a velvety look to it, whereas the whiter tan looking one is more of a woolly look, so it looks kind of like what a wool shirt would look like, whereas it's a little bit of a cottony look here in the center, but more of a woolly look towards the outside. So when we talk about the topography and the characteristics of mold growth, we are referring to how it looks as far as the smoothness of it or the roughness of it. So when we talk about something being varicose, it's describing it being kind of like a mountain-like. And so if you think about varicose veins and what they look like, if you've ever seen those, then that is describing this particular growth here, where it's kind of heaped up, kind of mountain-like, kind of a rough appearance to it. When we talk about something being unvenated, you're talking about it having a slightly raised center. So this one right here has a raised center, but the rest of it is flat. This at the bottom has an unvenated center, but then it also has a rugose type topography where you can see these little craters here, whereas this one over here is completely rugose. Now, microscopic preparation will vary based on the method that you're doing. So you could actually do a couple of different techniques. The one that I'm most familiar with and that I actually have enjoyed doing is using Scotch tape, and you take the tape and you kind of roll it across the top of the growth of the fungus. And of course this would be a mold type fungus. And what it's going to do is pick up some of those fungal elements, and then you would place that on a slide that has a drop of the lactophenol cotton blue, and you will tape that piece of tape down, and that lactophenol cotton blue helps to give a better visualization of those the vegetative form of the fungus. You can also do tease methods, or you could do a wet mount or a microslide culture, but a lot of it's just going to depend on where you're at and what their resources are and how much fungal cultures that they actually perform. Now we talk about the structures of the mold, then we are looking at the vegetative part of the mold. So when you do those different techniques to look at those molds on the slide, then you're observing what they look like. And some of the descriptive terms that you'll learn will describe this terminology here. When referring to sexual reproduction, we're talking about the different forms and ways that they reproduce. So Basidospore, you can see at the very bottom, and this happens with a little club-like reproductive structure that comes after meiosis. A zygospore in the upper left-hand part of the images, this is two round thick-walled spores that come together through fusion. And an ascospore is a sexual spore that forms within a sac-like structure. With asexual reproduction, you have the conidiophore, which is the structure that supports the conidia, and you can see that at the bottom. And then you also have the sporangium, which is a sac-like structure where the sporangiospores are formed. Other forms of asexual reproduction include chlamydia conidia. This is when you have swelling within the hyphae itself, and it can also give a clue to the type of organism based on where that swelling is. Blastoconidia are your basic budding of cells with a mother and a daughter, and this is how we typically see some yeast. Arthroconidia is where you have the asexual reproduction directly from the hyphae itself, with modification within the walls itself, so it kind of breaks off like little barrels. So again, this is an example of conidia, of what it would look like with the lactophenol cotton blue. There are arthroconidia, so you see those little barrel shapes where it has broken off. And the chlamydia conidia, where you see the swelling within the hyphae. And here you can see a sporangium with the sporangiospores within that sac-like structure.